Hello friends, it's me Rana Kumba Singh. In my previous lecture, I was discussing about the restriction enzymes. Let's see what is an palindrome. Palindrome is a group of letters which form words. If we read from one side or from opposite side, it gets same pronunciation. Let's see the example. Malayalam. If we read from this side, Malayam, it get pronunciated as Malayalam. If we read from this side, it also get pronunciated as Malayalam. It is now clear. Now see the example. 5 GAATTC 3 get paired with 3 CTTAAG. Okay, it got same pairing because guanine get paired with cytosine with 3 hydrogen bonds. Adenine get paired with thymine with 2 hydrogen bonds. Okay, it is clear. Now the restriction enzymes, it basically cut the DNA little away from the center of palindromes and it forms some sticky end on each strand. Okay, it is clear. Now see that stickiness of this is responsible due to enzyme DNA ligase. Also see this restriction endonuclease uses genetic engineering to form recombinant molecules of DNA. Now see the recombinant DNA technology. See, it is a foreign DNA or it is a vector DNA of plasmid. As I have tell you that restriction enzyme cutting the both foreign DNA and vector DNA at a specific point. Okay, see this foreign DNA has been cut from this point, vector DNA has been cut from this point. Now ligase join the foreign DNA to plasmid. It is the activity of DNA ligase. Okay, see both the point has been cut, it now joining by the ligase. This is the recombinant DNA molecules and it has been joined here. Okay, now this transforms into the E. coli. Okay, cloning as a host. Now this E. coli cell divide into two parts. Okay, see both have same thing. It is known as recombinant DNA technology. Now see separation and isolation of DNA fragment. It is basically done by gel electrophoresis method. What is a gel electrophoresis? It is simply that cutting a DNA fragment into a pieces using a restriction enzymes. Okay, it is known as gel electrophoresis. Now see this negative charge DNA fragment allowed to move toward the anode. Okay, under the influence of electric field, and this DNA fragment is separated according to their sizes using a agarose gel. It is now clear. Now the fragment which we have seen, it is get stained with ethidium bromide under the influence of UV light. Okay, it is now clear. See, it is a well, it is created here. A sample will be loaded here and electric field will be applied under the UV light. Then this particle start moving. This number 1 particles, it is basically undigested one, and 2, 3, 4 they are digested form. See 4 has been reached to this point. It is basically agarose gel electrophoresis showing a migration of undigested lane 1 and the digested lane 2, 3, 4. It basically recommend that DNA fragment purified this way is used for recombination. Let's see some questions. I will explain all the answers in my lecture. See, first question is that genetic engineering starts with the discovery of which enzyme. Okay. Second question is that which enzyme joined the DNA segment. Third question is difference between exonuclease or endonuclease. Fourth question is that difference between plasma DNA and chromosomal DNA. Fifth question is that explain gel electrophoresis. Sixth question is that explain palindromes. Also, अगर आपको हमारा लेक्चर पसंद आया हो तो उसे प्लीज लाइक करें, सब्सक्राइब करें और शेयर करें.